Okay, say cheese.
Or say twins. Hold on, wait, one more time. We can make a video while you're saying it.
I got invited to a celebration for Stanford's Twin Registry, and it was hilarious and fascinating. The Twin Registry became famous when Netflix launched their series You Are What You Eat. But the twins who came to the celebration volunteer for many studies, not just the one Netflix filmed. Only 8 of the twins were featured in the Netflix documentary, so it was fascinating to meet some who weren't with my camera rolling. Also, it's been about 2 years since the 8-week Stanford study that Netflix filmed, and the question on everyone's mind was,
okay, and now we do truly want honesty here. This is like the big question that everybody wants to know. After being in it, did your diet change?
I'll make an episode about all of that when I get back from Africa, but in this episode I wanted to focus on some twin-powered studies about immunity. There were 3 talks given at the celebration, one about the study Netflix filmed, one about the microbiome, and one on immunity. This episode features the immunity talk. I learned a lot from it and thought some of you would like to see it.
She is twin A, so.
Twin A, so you're twin B?
That's right, that's me. Yeah.
What does that mean? Was she first born?
She's first born, yeah. On the birth certificate.
On the birth certificate,
yeah. Twin A or twin B.
But she's a minute or 2 older than you?
30 seconds.
30 seconds?
Yeah, that's
wow.
it.
Your mom had them fast.
She did. Yeah, and Mary will never let me live it down.
Oh, yeah. She's always second place.
Yeah, big sis.
We are going to have some fun tonight. What a hilarious audience. Wow, I wonder how many people get to look out at something like this. My name is Christopher Gardner. I have a PhD in nutrition science, and I'm going to be a moderator tonight. Welcome to all of you. We have a lot of people to thank for putting this all together. We're going to start and kick this off with a god on the Stanford campus, Mark Davis, born in Paris, raised by wolves, I clarified that before, is largely responsible for this, for his management with Michelle of the Twin Registry, and he has an incredible background. So I'm going to introduce him quickly. It's too long to go on and on, but I just picked out things like he's in the Institute of Medicine's National Academy. One of his favorite awards is being in the Royal Society of London that Isaac Newton and Ben Franklin were part of. He's a professor of immunology, and he created this human immune monitoring center where when we do studies, my group does nutrition studies, we're always trying to look at what's happening to immune function and inflammation. He has this incredible center that's just been growing and growing and building over the years, which I learned for the first time was started on a shoestring with $1.7 million, if that sounds like a shoestring, and created such an incredible facility of methodologies and equipment that the Gates Foundation then gave him $50 million to build this out on campus. So Mark has just an incredible legacy. So without further ado, he has a slide deck and he's going to talk.
All right. And so the very first thing I have to do is thank Michelle King, who many of you know. We call her the twin whisperer of Stanford. I spent the first 20 years at Stanford doing molecular biology, understanding some nuts and bolts and some very basic things about how T cells recognize things as foreign, and it was a big sort of empty spot in our understanding of the immune system. That worked out pretty well. And I started thinking more broadly, and then I got this job as in the institute director, and I said, well, I really have to think broadly. You know, how can I do something that's just beyond what my lab can do, which is pretty good, but you can't do everything. And to just think more broadly, what is the next level of immunology? What can we do that goes beyond what anyone can do at this moment? And that involved the realization the immune system is a major life support system. It's involved in many diseases and more medical interventions right now. We have infectious diseases, autoimmunity, cardiovascular diseases, cancer, neurodegenerative diseases, aging, vaccines, organ transplant, and probably some I've forgotten. And then just what is an immune system? It's really a bunch of cells that talk to each other with over 100 different soluble molecules, like hormones, although they're not called hormones. And there's just lots of different flavors. It's all the white blood cells. So we try to get rid of, if we get blood, we get rid of the red blood cells because we don't care about red blood cells. They're there, but you know, We don't think they're important. It's the white blood cells that are the immune cells. And they come in different flavors, they have names. I often tell people that whereas molecular biology came from physics and chemistry, that immunology came from witchcraft and shamanism, where the important thing was to know the secret name of something, and then you gain power over it, right? So we say, oh, it's this very special T cell, you know, and no one else knows the name, but I know the name, so I'm very powerful. Yeah, it's really, there's a lot of flaky stuff like that. But just for general entertainment, though, I have to show you one of the more spectacular things, which is a T cell attacking a cancer cell. And it's attacking cancer cells with these special cell poisons, which are labeled in red here. And it's really quite an amazing snuff film here where, okay, it's now recognizing, and it's shooting these poisons into the other cell, and they're bursting and spreading the poison and killing the cell, clearly. You can show your kids this if they're old enough. So anyway, the immune system is really important, but it's so complicated that it's been treated essentially as a black box in medicine. If you go to a clinic here or anywhere else and say, I'm worried about my heart, they'll be all over you. They'll be drawing blood, they'll be throwing you an MR machine, they'll be giving you all sorts of prescriptions. If you ask, how's my immune system? They'll look at you like you're some kind of nut, you know, kind of like, what, are you sick? Well, but that's not the point. The point is we should be better. We should know more about this than we do. If you think we know a lot, then you should just see what are the standard assays. Basically, white blood cell counts was developed in 1915 as a diagnostic for, you know, if you have too many white blood cells, that's a problem. There are things that happen there. And the biggest advance recently was 1959, where something called complete blood cell counts, which is a slightly better defined white blood cell count. But meanwhile, since 1959, there have been 15 Nobel Prizes awarded in immunology. So there's been huge advances in our knowledge of the immune system, but it just hasn't made it into medicine. Why hasn't that? It's a really strange thing. And we know you want immunology, you know, because people buy this stuff. It's meaningless. There's no studies, there's no science behind these things. But you go into any pharmacy, you'll find like a whole bookcase full of immune boosters. And this one is particularly insulting. This was created by a school teacher. And I have the greatest respect for school teachers. Some of you may be school teachers, but this is the science I'm talking about. This is something I've spent, 45 years or so of my life trying to understand. And I don't think school teachers really have much expertise in this. But it doesn't mean they can't sell something. But the whole puzzle of why don't we have a better understanding? Why don't we have a cholesterol test for your immune system? That you can just give a blood sample and they can say, oh yeah, you're good, you're doing great, or no, you're in real trouble here and you should really get your vaccines and maybe some other stuff. And the problem is that when you hear a lecture about science or you read a textbook, it's all wonderful. It's all inevitable progression to glory and understanding and enlightenment. They don't tell you about all the blind alleys and the mistakes that people make. We're human beings. We have ideas. We think this is a good idea. We pursue that, but maybe 20 years later we realize, that wasn't such a good idea. We really should have done this. And I think we're in that situation with the immune system, and why it hasn't progressed that much in human beings is that we're in love with mouse models. We're the first to use inbred mice 60 some years ago. It's been a great tool. They do whatever you want. They don't talk back. They just sit there. And partly because they're inbred, so they're really dumb actually. And wild mice bite, so no one wants to work with wild mice. But inbred mice is good. And if you think inbreeding is good for you, you should just look at the crowned heads of Europe. That inbreeding has not really worked out that well for them. They're also dumb mostly. Anyway, so the mice have been the go-to system. 40 years ago, someone said, I don't know who, that the way to advance human disease treatments is to make a mouse sick with a particular disease like humans, like autoimmunity, like cancer, and then figure out how to cure it. And then this will be the way forward in terms of treatments. That has worked almost none of the time. Few things, definitely a few things. But, and this led Pedro Lowenstein, a friend of mine, to say to his medical school class, for the mice in the audience, I have wonderful news. We can cure your cancer, we can cure your autoimmunity. When you can be that sarcastic about something, you should realize there's some problem. We should be maybe doing something different, or at least thinking about doing something different. And so that's what I thought. I thought, hey, the mouse model, if the mice are not telling us enough information, we need to look at humans. And not that it's easy to look at humans, it's actually really hard, and it took a lot of work to get where we are today. But back in the day, 15-ish years ago, I offended a lot of my colleagues by writing this paper and saying that these mouse models of disease were not really working that well, in fact, mostly not working at all. The only way we could get out of this is to start looking at humans and to do things seriously. But at that time, people thought human work was just descriptive and led nowhere, and there were very few technologies that you could employ on humans. And so it was a big challenge that most people decided to ignore, but we didn't. And that's where we got to work, and that's where the Twin Registry, originally at SRI, now at Stanford. Peter Brodin, who's a Swedish postdoc in my lab at the time, now he's a professor at the Karolinska and other places. And the whole team here, including Gary Swan, who really invented the Twin Registry at SRI in the 1990s. What we were doing here and why it was a big hit was that previously people were correlating genetic attributes, polymorphisms, with particular things like immunology, but there was no context. It wasn't like genetic is the big influence here versus environment. It was like, hey, genetic, there's this little association with genetics. But what we could do with the twins, and some of you may have been part of this, I think, we were able to actually look at the whole picture of how much is environmental, how much is genetic. And you can only do that with twins. And so what we basically set out to do was to define sources of variation in the immune system. We know people's immune system is very highly variable. Is it mostly genetic or is it mostly environmental? And so we analyzed the blood samples of 210 twins from originally the SRI registry, ages 8 to 82, over a 3-year period. We measured more than 200 variables, cell types, serum, cytokines, vaccine responses, and so forth. And we're greatly, thanks Christopher for the shout out to the Immune Monitoring Core facility. This has been a very innovative, very useful thing to basically kind of an industrialize immune assays, because you need to do a lot of them. And it's not a good thing for your students to be doing. But it's great to have a facility, and you and others have recognized that. And I'm particularly proud of our motto, Immunology for the People. Sometimes people ask me, are you from Berkeley? Are you really, what the? Here's some of the, I'm not going to torture you with much data, but I think it's pretty simple. We looked at a lot of things, these are different cell types. All you need to look at are these dots. So if a dot is in this column for this cell type, it means there's no obvious genetic influence. And then things out here means there is some influence. And all it is to say, is person X more similar to their twin than they are to other people? And if the answer is no, then there's no genetic correlation to speak of. If you are more similar to your twin in some variable, like for instance, maybe this cell type, then yeah, there's something genetic there, something inheritable. But what you can see is that we found 70% of all these variables, there was no genetic correlation. You would be no more similar in this particular variable than your twin. And if you like these sort of graphs of interactions, these are purple. All you need to know is that purple is no genetic influence, and yellow is there's some genetic influence. So you can just see there's a massive amount of probably environmental influence and not genetic influence. And it makes sense in terms of this is a sensory system. We have other evidence that says T cells can see as like a single molecule of a foreign antigen. And a sensory system is something where you, like your nervous system, you land someplace on Earth and you have to learn the language, and you have to eat the food, you have to experience the diseases of that place, otherwise you're in big trouble. So you just do that. That's what we do. So your nervous system does that, your immune system does that. It makes some sense. And then there's some cute things that we showed, like we found that young twins are more similar in some things than older twins. Consistent with the older you are, the more environmental influence you have. That can make a difference for some things, although not others. And then there was a really strange thing. It turned out that we had, I think, 15 monozygotic twins where one twin had a cytomegalovirus infection and the other twin did not. And so that we can just look here, and we found out that, so cytomegalovirus is a herpes virus that about 50% of the people in the US have, much higher percentage in third world countries. And it basically remodels your immune system. And this is the evidence that it's doing that. It doesn't make you sick unless you have an immune deficiency. It's kind of the perfect virus. And it's even more perfect because it inhibits other viruses. We found that people that had CMV actually gave a better flu vaccine response than people that didn't. But it's a virus, so it has its own agenda. Mostly you don't feel it, you don't experience it, unless you're getting some immune deficiency like a bone marrow transplant or something where you're irradiated. That's not the problem. But anyway, so this is all part of the scheme we've had in the immune monitoring. In the Immune Monitoring Center is a big part of gathering lots and lots of different data sets to try to understand human immunology. And the twins have been a huge part of that. I should also mention we have over 1,000 citations for this paper. That is a huge paper. Most papers have like 3 or 4 citations in their entire life. This means that it had a big impact. It's in a big journal, Cell is one of the major journals. And this was also really important in human immunology because it said this is real science, that we're doing something here that you couldn't do in a mouse. The inbred mice are kept in the basement wrapped in a heap of filters. They don't go anywhere. The only exposure they have is to graduate students and postdocs. It was a breakthrough paper because up until that point, people were dismissing human immunology as being like this trivial thing that, well, yeah, right, you couldn't do mice, so you did humans. We had some very good papers, but they were turned down by the big journals because of this prejudice against humans, which is a little strange. This was a breakthrough paper for the Institute, for my lab, for human immunology generally, because it kind of said, oh, here's a top journal, and they actually have this, and it's a very meaningful study. So, and as the citations also support. So it also was important for me because it was the twin studies was something I proposed in 2008 to support a grant renewal that most people thought the grant would not be renewed. But because I had this proposal about a twin study in it, the grant did get renewed. And it's now been renewed 5 years, now 3 more times, including just recently. So again, this kind of, if you can't get the funding, you can't do the work. A more recent use of the Twin Registry has been to address a really important problem in flu vaccination, because for more than 60 years, people have noticed that even you have multiple flu strains in a flu vaccine, most people only respond to one of those strains. They have a favorite strain. And a very influential idea about why you have a favorite strain, and a very original name, was called the doctrine of original antigenic sin. You can tell what sort of education that Thomas Francis had. The concept is that you have a favorite strain because that's the first flu strain you were infected with, and that biases your immune system, and ever after you just like that strain and you tend to ignore other strains. And it's generated a lot of colorful review titles, particularly, and including this title, The End of Sin. What's amazing is a postdoc in the lab, Vamsi Malajosal, who's worked on flu basically all his life, challenged this idea and thought, well, I'm not sure this original sin thing is correct, even though everyone kind of talks about it. There's no critical tests in humans. And so he used the Twin Registry and the twins that we had vaccinated with flu to confirm that, yes, indeed, twins, if one twin has a preference for a particular flu strain, then almost certainly the other twin will have that. But more importantly, because twins are raised together, they'll get the same infections at the same time. It doesn't distinguish a genetic principle versus an environmental one. So what he did is he then tested a whole bunch of other strains in addition to the ones that were popular to say how similar are twin pairs to each other in terms of historical other flu strains. And turned out to actually mostly not. And so he was able to estimate that only about 20% of this strain bias, it's a bias of 70, 80%, but only a fraction of that could be attributed to this original sin idea, and that it really had to be something, it had to be genetic. And he's followed through, I won't show the data, but we have I think, really spectacular paper that hopefully will get accepted someday soon, that will put flu vaccination on a whole other basis. That it is genetic and it is actually class 2 MHC polymorphism for anyone that might understand that. And he tested a vaccine formulation to test this idea, and it works great. a big problem with flu vaccine. One reason it's not a very good vaccine is the strain bias phenomena, because you get a vaccine with 4 strains in it, but if you only like one and you get infected with one of the other ones, you will get sick and you might die. That's why flu vaccine is not as good as the COVID vaccine in terms of effectiveness. So anyway, this work, without going into the details, this work is fundamentally changing how we think about strain bias. And it's not just going to be for flu, it's for other things as well, as a general principle. So anyway, so that's all was made possible by the twins and the registry, so thank you for that too. Lastly, because of the success we've had in twin studies, we were able to get another major NIH grant looking at aging now in twins. I bet all of you are aging, and I hope gracefully. I'm trying, but I don't have a twin to compare with. But anyway, Shai Shanor, a former postdoc who actually helped with some of the early twin work, but now is a professor at Technion in Israel. We have also access to samples from Project Baseline, which is a Google-funded cardiovascular study. And so we're looking at the influence of the immune system on cardiovascular health. And it's really some unique samples, some unique things. And so we're hoping I tell cardiologists actually not completely seriously that cardiology is a sub-discipline of immunology, because it's about inflammation and white blood cells and stuff like that. But I don't think they take that really that well. So I have to thank people in my lab who dress alike. I'm not sure why. And they're just fantastic group of people to work with. And thank you, twins.